Good morning to all of you and welcome to day 2 of this workshop. So the first speaker of the day is Professor Helmut Grubmüller, who is honorary professor for physics in University of Göttingen and also a director at Max Planck Institute for Multidisciplinary Sciences. He heads the Department of Theoretical and Computational Biophysics. His research, is to look at the physics of biomolecules, proteins, protein complexes, DNA, RNA complexes, and so on. So we welcome Professor Helmut Grubmüller for his talk.
All right. Good morning, everyone. Can you hear me well in the back? All fine? Great, that looks good. I rarely get so many thumbs up, thank you. So yeah, we are getting into theory now. Ooh. But after all, this center is a theory center, right? So I think that's quite fitting. Can you give me a show of hands? Who of you is a biologist? Just to get a bit, okay? And some physicists there? Oh yeah, great, hello, good morning. Chemists, computer science, philosophy? No, okay, great. So I'll try to adapt as good as I can. There will be a little bit of math, but no worries. There will also be quite a bit of intuition. I thought I share with you kind of 4 stories. And of course, that's not my work. That's the work of a wonderful bunch of people here. I should say the department I'm heading hosts 3 groups. And obviously, I mostly report on work done in my own group. That's about 1/3 of these. But I should also emphasize, we always welcome bright people. The main research interest or the main goal which we have is actually, we want to understand how protein works. So that is a bit of a contrast to what you guys often want to do. You want to find proteins who do some function, don't care so much about how precisely the internal mechanism is, but that is our focus, right? So we want to understand how this, like to call it nanomachines, actually work. What's the principles? And we continue to be surprised because whenever we try to find out some functional mechanism, of course, beforehand you think about how would you do it if you were an engineer, and typically nature does it completely different and mostly better. So that is one of our driving forces. And as you all know, most of the biochemical functions proteins do and other RNA-based machines, like we'll get to the ribosome shortly, most of it is driven by structural changes, conformational dynamics, conformational motions. So ultimately, we want to understand how conformational motions implement biomolecular function. And as you also know, specifically on fast timescales, these motions are really hard to observe. And that is why we think, as theoreticians, we can help a little by computer simulations of these motions, which are based, all of them, on fundamental laws. And so before I share some stories, we have seen a little bit of molecular dynamics already, also on the posters. I'm glad to see that. But I think it might be a good idea to first introduce a little bit the method, and then I'd like to illustrate to you what we can do with it. Yeah, just to give one more motivational example, that's one of my favorite machines. Most of you will know that's mitochondrial F1 or ADPase. F1 is the upper part here, and it is literally a car engine, right? It works like a crankshaft in a car with moving cylinders in the pistons here. It's the same principles, except it synthesizes ATP by turning around this stalk here. And again, there's actually really conformational motions which perform the task. So specifically what you find here is, if you look at it from the top 3 binding sites, one ATP loaded, one ADP loaded, and the phosphate, one empty. And it is the rotation of this asymmetric central gamma subunit, the stalk, which actually changes the conformation of these binding sites such that they have differential affinities to ATP and ADP respectively. And so this thing is just ready to expel the product ADP and ready to accept a new, on the empty side here, a new ADP and a phosphate, while at the same time with a new rotation, the synthesis of the ATP to ATP takes place. And the mechanical energy provided by this rotation is actually used to drive the synthesis. So this is a mechanical chemo energy transducer. Now, in contrast to my car, this thing can work in two directions. It works typically in synthesis direction in your mitochondria, but you can also, and people in Japan, Kinoshita and others have shown that marvelously with optical tweezers, you can also fuel it with ATP, and then the central axis will be rotating like an electromotor, so like my car engine. So I tried very hard with my car to push it and never saw some new fuel coming out of the back. So this machine can do that. And the reason why it can do it, it is near 100% efficient in contrast to my car, right? My car is like 25, 30 if I put effort into it, but that's it. Here we are near approaching 100% efficiency. That's the reason why it's also reversible. We would like to have that in our engine. So that's also one motivation why we try to learn how that works. And of course, you know this guy, ribosome. My first story has to do, centered on the ribosome. So let's try to get into that. I should really emphasize again that our simulations are based on first principles, meaning that we start from quantum mechanics and then work our way upwards from the bottom up. That is, when I'm talking about modeling, or when you guys talk about modeling, that's a totally different thing compared to what you typically are doing. You try kind of observe something, and then you try to come up with a model which fits the observations and ideally makes some predictions. Here it's just the other way around. We just build from fundamental physics laws upwards. So our model is without biological observations. Of course, it rests on fundamental physics. And let me illustrate that a little bit more with an example which we used quite a while ago. This is aquaporin, and it's in, for example, in your red blood cell. It's also in your kidneys, in your brain, in your islands, and it conducts water. That's the whole job. It's a membrane protein. It's embedded in a membrane, and it passively conducts water at an incredibly high rate. So it's about 1 water molecule per nanosecond, which is, by the way, also what brought us, made us interested in this system, because as you will see in a couple of minutes, the main limitation of our approach is timescales, right? So if you want to cover 10 times more microseconds or something, you have to spend 10 times more computational effort. And so our timescales are very much limited to the microsecond range, but that's not so bad. Most biological function actually on the molecular level happens at that timescale. So that's not so bad. You may know that protein folding occurs on a millisecond or so timescale. So for most protein, that's still out of reach, but biochemical functions are, many of those are there. And speaking of nanoseconds, obviously we are right in the right ballpark. So that is one motivation why we looked at that. So how does it work? First of all, we start with typically with a known structure, put that into the computer, by which I mean every single atom is actually put in an array XYZ coordinates, and we tell the computer where every single atom sits, not only of the protein, but also of its environment, like a membrane here. The colored thing is the protein. The membrane, you can see the lipid tails in green and the heads in yellow, and also the water. You better include water if you want to simulate water permeation, right? But also very generally, we need to have the water in the simulation box, as we call it, right? That's our simulation system, because the water exerts quite a bit of effects on the protein. Proteins in vacuum don't work typically. So we need that, despite the fact that we spend quite a bit of computer time on just calculating water. Also, to give you a kind of perspective, the amount of computer time we are using in terms of energy every year is about the amount a Saturn V rocket uses per launch, right? So that's about the energy which we need to drive our computer, which are quite a bunch of GPUs. Okay, so once we have all the atoms in the box, we calculate the forces between them. And that's now the hand waving picture. I have a more rigorous picture for the physicists and mathematicians and computer scientists amongst you later on in a couple of minutes. But the hand waving picture would be, because most of the atoms carry partial charges, they will interact with electrostatic forces on top. So we have Coulomb interactions between them, and that means that if you want to calculate the force on every single atom here, you need to calculate the force between this atom and all the other atoms, at least the partially charged ones. And that gives you a total force on this atom, and then you do the same on this atom. That's kind of a party handshake problem, right? So you have a big party, in this case of 100,000 people, and if you want to handshake each with each other, that will be a long party, and that's exactly why we have to spend so much computer power. It's the electrostatics, which is quite a bit of an effort. Again, for the experts, there are fast methods around, and we are using them. Particle mesh eval and fast multipoles are the keywords you might want to look up. On top of that, we have van der Waals interactions and Pauli repulsion. Atoms don't want to move through each other. Lucky for you guys, otherwise we would all fall through the floor. And then we have chemistry, right? So we have the chemical interactions here indicated by springs. I do know that chemical bonds are not springs, but it's a pretty good approximation. And also all these forces we need to calculate as a function of where the atoms currently are. And as a result of these forces, we go back to our good old friend, Sir Isaac Newton, and ask him, what can we do with those forces? Well, they accelerate our atoms and change their velocity. And the changed velocity will change the position of every single atom in a very short time frame. Here we talk about femtoseconds, 10 to the minus 15 seconds. And after that time, all the atoms have been moving a little bit. They changed their position a tiny little bit. And so do the forces, right? So if the chemical bond stretches a little bit, the force will change. And so we have to recalculate these forces again, the whole bunch of work again. And that again changes velocities, and then you can see where that leads. We have to have a big loop and repeat these force calculations all over again and over again. And if we accumulate 1 million, or typically these days 1 billion of these loops, which we call integration steps, because we integrate numerically the Newtonian equations of motions, then we end up with a kind of a movie which shows you in real time, if you want, not exactly real time, it's super slow motion. We see a picosecond about per second here, per second movie. And you'll see then the motion which is going on. And I'll show you in a minute, we have good reason to believe that this is pretty close to reality. So this is how a molecular fish would look at the system. What you can see nicely is here the protein again. You can see the membrane, which is not as ordered as you see it in old textbooks. It's quite a chaotic system. So the lipid tails, they are quite disordered. And then you can see the self-diffusion of the water molecules. That's what your coffee about looks like if you zoom into it. And nicely, you also see the water molecules here passing by, right? So through the channels. You have to wait a few minutes in order to really see the passing by. We are still too fast here. But in this case, what we did is we counted the water molecules going through per nanosecond or per microsecond and compared that count with measurements. So measurements of the permeability of the water through the channels, and that agreed pretty well. Moreover, we were analyzing which amino acids the water molecules were mostly interacting with, suggesting mutations, calculating, predicting what should be the effect of a particular mutation on the water permeability. That was a prediction, and that was also confirmed later on experimentally. So these are the reasons why we think that our simulations are not so far away from reality. All right, I was promising for the experts, just very briefly, my students in my lab, they really need to know what they are doing when they carry out these simulations. And so just in a very brushstroke way, ideally, you want to look at the Schrödinger equation and solve that, which is an equation which tells you how the psi functions, quantum mechanical wave functions, develop in time as a function of the positions of all the nuclei, the capital R here, and all the electrons. So just imagine This R, if you've got a system of 100,000 atoms, that's about 1 million electrons. So 3 million XYZ coordinates. This is this innocent little r. And the same for the capital R, all the atomic positions, the nuclei positions. So the equation is pretty simple. You can write it down on a sheet of paper. But to solve it, even with the fastest computers these days, just impossible, except you only have 2 or 3 atoms. So we have to do drastic approximations. And one of the central approximation, physicists know that, is the Born-Oppenheimer approximation, which tells you because the nuclei are much heavier than the electron, they move much slower. That's also true for quantum mechanics. And so what we can do is we can pretend that the nuclei, just for a little bit of time, remain stationary and just look at the electrons, how they react as a reaction to the electrostatic field of the nuclei, and what ground energy level they adopt to. And this is actually this equation here. So this is now a different Schrödinger equation, the time-independent one, much easier to solve. And notice this tiny little semicolon. It means that now the nuclei are not dynamic variables anymore, like up there, which makes it complicated, but they are just parameters. The game here is you give me the positions of the nuclei, and I calculate you with this equation the ground state energy and the psi function, the wave function of the electrons. Much, much easier, yet still too complicated for such a large systems which we want to study. And so as a second step of approximation, what we do is we kind of tabulate the ground state energy. It's a function of nucleic position, so it's a very high dimensional function, but we devise an empirical force field looking like that. And those who do molecular dynamic simulations, they know it, of course, that there's all these crazy terms like bond, angle, and electrostatic van der Waals, you name it. These are all simple functions which approximate a function given here by the ground state energy of all the nuclei, which is non-analytically, right? You can just calculate it numerically, but the approximation, it's analytically, and that makes us, gets us into the game. Now, for those who are into AI, when I say we need to approximate a high dimensional function, that should ring a bell, right? Because this is what deep learning actually does, right? Whatever you do, any neural network, essentially it approximates a high dimensional function. And that's why it will not come as a surprise to you that many groups, including our group, also work hard these days to replace this force field, this analytical force field, which is a SOLA approximation, with a deep learning force field, which then would hopefully be a bit more accurate. Actually, I should also say it is this force field approximation which creates most of the trouble. So if anything goes wrong in a simulation, most likely the force field is to blame for. I was promising 3 approximations, and the third approximation actually is that we now do not treat the motion of the nuclei in this force field now as quantum mechanical motion, which it of course is, but we approximate it, as I mentioned, with Newtonian equations, that is with a classical approximation. And you may ask, what the heck is this guy doing? We all know that atoms are intrinsically quantum mechanical atoms, objects, but it turns out that for biological systems, that means at room temperature, in condensed phase, not in vacuum, where you all have these bumps by the solvent, which actually dephase the coherence of the quantum mechanical wave function. Under these conditions, this approximation is pretty good. There's a few things you can't do, for example, calculate the heat capacity, but other than that, this is a pretty good approximation. So we have three approximations: Born-Oppenheimer, fast electrons, slow nuclei. We have the classical approximation of the nucleic motion. By the way, those who want to go into that field, this is often mixed up. The electrons we treat fully quantum mechanically. So if you say molecular dynamic simulation is a classical approximation, that is only half true. It's only true for the nuclei, not for the electrons. That's a mistake many make. Okay, and then we have the force field approximation. So that is in a bit more detail what we are doing when we carry out such a simulation. And I've already given you a bit of evidence how we test our simulation with this water permeability. There's another test which I like very much and which I like to share with you here, which we did actually quite a while ago, where our simulations were way too slow, but we repeated it this time, and I'll show you how that works. So this is an atomic force microscopy experiment simulated now with molecular dynamic simulations. And again, for the experts, we simulated atom by atom. So this is just a representation, which makes it easier for us to see. And of course, the whole thing is solvated in water, which we also don't show in the graphics, but we have it in the simulations. And what we do here, this is streptavidin, and we drag out the ligand out of the binding pocket. And this is really exactly as it is done in an experiment, in an atomic force microscopy experiment. The only difference being in the experiment, they only measure the peak force. So here I plot the force as a function of how the simulation proceeds, by which this kind of virtual spring, in reality, it's the cantilever of the atomic force microscope, actually pulls here. And in the cantilever, you don't have time resolution, not much. And so you only can measure the maximum force, whereas here we have the whole force development, the whole force profile. But of course, what we can do is to compare our maximum with the measured ones. And this is actually what comes out of such a simulation. That inset gives you an idea how the real experiment looks like. That's the force, the cantilever of the force microscope. It's a tiny little spring which bends a little bit as a reaction of this single molecule force here. And it is this bending which can be sensed with a laser pointer, essentially, and measured. So you can measure piconewton forces that way. And what I'm showing here in this graph here is the force both measured in the AFM experiment. These are the blue dots here, the circle, and the ones which we calculate from the simulations, that are the green ones. And the x-axis here, note the logarithmic scale here, that is how fast we pull, right? The loading rate. On the right side, we pull extremely fast, so the whole rupture process is completed in a few picoseconds, and here in minutes or hours on the left side. And as you can imagine, for the experiment, it's pretty hard to get faster and faster. The speed by which you can retract the cantilever is actually, and measure the force, is actually limited by the resonance frequency of this cantilever. So you want to make the cantilevers as small as you can. And Simon Scheuring, with whom we collaborated at Columbia, he's a master of fabricating very small cantilevers. So he can do the fastest experiments, pushing here to the right, up to there. And from our side, as I mentioned, if you want to simulate 10 times longer, you need 10 times more computer power and calculation, computer time. And so this is a very easy one. And here it gets harder and harder. And such a point, this point took us a month or so to calculate this point. But as you can see, the overlap is pretty good. And I should emphasize that there is not a single fitting parameter in there. That's a consequence of the different model building, which I was mentioning before, right? Your models, which you are typically using, they need fit parameter, and you fit them to your observations, to your experiments. But here's because it's building from fundamental laws, there is no fit parameter, except if you want to say H bar is a fit parameter, but that would be a bit unfair. So there's not a single fit parameter in there, and yet you get this nice agreement. And we have seen that over and over again, which is one of the reasons why, again, I think we are relatively close to reality. All right, trying to move on. So let me get to the 4 stories I want to share. And I'm not sure if we managed to complete all 4, but let's start probably with the ribosome, because that is probably closest to your heart, I understand. And so let's go into that. This is work mostly by Lars Bock in my group and his co-workers. So I don't need to introduce the ribosome, of course. tRNA is coming here to the A site, delivering the amino acid to the growing nestin chain, which is being pushed out of the nestin tunnel, but only, of course, if the cognate nucleotide fit nicely here. This is a cryo-EM picture, and I'm showing that because it shows an antibiotics. So here, now you have to mentally rotate the ribosome a little bit. Here is the A site again, tRNA coming in. Here's the amino acid. And the red thing here, this is an antibiotics, erythromycin. And the question was here, how does the antibiotics actually, in this case, stall the ribosome? How does it work? Obviously, we would like to know that, whereas I should say, for most of the antibiotics, we actually don't know how they work. We just know that they work, but not how. And Of course, you might say, and that was also the widespread thinking in the field, you might say, well, that's pretty easy. There's a narrow tunnel. The nests and chain has to be pushed through the tunnel. If you plug a cork like this molecule right in the middle of the tunnel, you block this tunnel and that stalls the ribosome. Easy, right? Except people knew for a while that it's sequence specific. So certain sequences stall here and others don't. And actually you can see, if you look very closely, the green nests and peptide here already is about to pass this red antibiotics here. It's about here already. So it gets stalled here, but not like a cork in a bottle, right? And again, other sequences don't get stalled. Now, obviously, we have this nice structure here. We don't have a structure of the non-stalled situation, right? Because, yeah, you can't make a structure of a running machine. And so the question was, what's the difference now between the antibiotics present in the tunnel and absent, and how precisely does it now perform the stalling? And of course, what we see when we look into the tunnel here from the simulations, here that's the antibiotics here, and this is symbolically the nests and chain in terms of amino acids, we do see a number of enhanced interactions to the tunnel walls here by the amino acids as compared to the situation where we removed in the simulations now the erythromycin. So in the simulations, you can do that. In reality, it's a bit hard. Well, you can also do it, but then not take a structure anymore. And so we see more interactions, but that does not suffice to explain the stalling. The interactions are just not strong enough. There must be something else which actually performs the stalling. And what we saw, quite to our surprise in the simulations, was, as indicated here, part of the ribosomal RNA here, forming the tunnel here, gets a structure, sees a structural change in the presence of the erythromycin. And this seems to be quite a short range interaction, but it goes over 20 angstrom, 2 nanometers. So that's quite a long range allosteric change just due to the presence of this erythromycin. So it changes the shape of the tunnel walls. So you might say, okay, what that? Well, that's mostly phosphate pointing here towards the PTC, which is the active site where the amino acid comes in and gets, by nucleophilic attack and the peptide bond formation, gets attached to the growing chain. So here you can see the amino acid coming in, or here. And that would be the arrow indicates the nucleophilic attack where it needs to be attached. This is at the active site, the PTC. And now what we see actually is that due to this retraction, this distance becomes a little larger. And let me go a bit into detail. So this is a zoom into there. That's the phosphate backbone. And the superposition shows in red the structure in the presence of the erythromycin, and in green in the absence of erythromycin. You can see the structural change now in pretty much detail here as we get it from the simulations. Now, here we have the negative charge of the phosphates, and here we have an incoming lysine. And it's been known that if you have a lysine at this position, then stalling is particularly pronounced. Okay, so it seems to be, that's what we see in the simulation, pretty easy. We see that it is this negative charge which attracts the positive charge of the lysine and just drags the lysine with it. What we plot here on this graph here is the position of the backbone as a result of the erythromycin presence, here with, here without, so the distance is larger with erythromycin. And here it's the distance between the backbone here and this asparagine. So that also increases, which tells us the asparagine mostly stays in place. And here we have the distance between the asparagine and the lysines, so this distance, which tells us, yes, indeed, the lysine is kind of pulled away, and the distance between the lysine and the asparagine actually increases quite a bit. And so we thought maybe this is the reason why the nucleophilic attack cannot proceed anymore. It's just too large a distance. And actually the distance turns out to be significantly larger than those which we know from most unperturbed X-ray structures without erythromycin. All right, how can we test it? Well, first we test it in the computer by replacing the lysine here with an uncharged alanine. And lo and behold, if you do that, you don't see much of a change anymore in the presence and absence compared to each other with and without the erythromycin. Now, again, the distance between the alanine and the part where you need to do the nucleophilic attack. So here the stereochemistry seems to be largely unperturbed. If all that is true, then it seems that we need a positive charge there for maximum stalling efficiency. And apparently a lysine would be a good choice, actually the only one else. And so we predicted that lysines should also affect stalling. And strangely enough, going through the literature, nobody has looked at a lysine at this position at that point in time. And so we went to our friends and asked them to measure that. And indeed, if you have here lysine or arginine, you see the maximum stalling, and others don't stall so much. So that also confirms our prediction here. So that seems to be the main mechanism, actually, a combination of two effects. One, which I didn't dwell too much into, that is interactions which are strengthened just by the fact to the walls here, just by the fact that the erythromycin is pushing the nascent chain against the wall. We have stronger, mostly van der Waals and hydrophobic interactions. But the main effect, and also the one which makes the sequence specificity, is actually here the perturbation of the active site in the presence of the erythromycin. And this was a while ago, and since then, in a number of collaborations, we have looked at different stalling mechanisms, both with other antibiotics, but also stallings. There are certain sequences, SEC-Y would be one SEC-M, where the nascent chain stalls just by itself without any other molecule interfering. And we have looked also into those. And the common theme is that actually we stall by perturbation of the active site via allostery. So we rarely see stalling just by clogging up in the exit tunnel. We mostly see stalling by allosteric interaction back to the PTC, to the active site, which seems to be super sensitive to any perturbation. If you perturb it just a little, it slows down dramatically. And this seems to be the main mechanism of stalling as we see it here. And because of this allosteric interaction between pieces quite far away already progressed in the tunnel and the PTC, we got quite interested now these days, and this is ongoing work, to actually simulate how the growing nascent chain is pushed here to the right through the active through the tunnel, through the exit tunnel. And mind you, it's not pulling, right? It would be easy to understand how you can pull the thing out. And actually, as soon as it peeks out of the tunnel, there are enzymes which actually grab it and pull it a little bit. Co-translational folding would be one keyword you might want to look up. But until it traversed this several nanometers through the tunnel, it is just pushing. And the persistence length of this nascent chain is super short, just 1-2 amino acids. So this is a highly flexible polymer, and it is as if you want to push a spaghetti through a winding tunnel, right? And I imagine that's a hard thing to do, and so we got pretty interested recently how the physics behind that actually works. Some of our colleagues argue, well, the widening of the tunnel gives a little bit more entropy, and the entropy helps the system moving out, but there's a constriction region quite over there where two proteins actually meet the tunnel. And it is really a riddle still today. Nobody knows how it works that the ribosome is able to push through this tunnel. Apparently, timescales are pretty critical here. So if you work on that, if you want to work on this project, you also need a little bit statistical mechanics because, as you might know, the ribosome manages to attach about 20 amino acids per second. So we are on the millisecond timescale at best. And currently, to carry out, like, let's say, 10 microseconds costs us a couple of weeks of computer time, right? So we cannot do these simulations in real time, and we need to invent a few tricks, and there are a few around, and we need to refine them and apply them in order to accelerate that artificially on the computer for the experts. Biased molecular dynamics would be the keyword you might want to look at. And then this is how we do these simulations currently. All right. let me look at the time.
Sorry, do they also explain the resistance mechanism?
These simulations?
Yeah.
We haven't put antibiotics into these simulations. All we have done so far is started with the nests and chain already in the nests in the tunnel, like we get from the structure, and removing the erythromycin, and then see how the system reacts. That's easier. This one is the hard one. We haven't managed to really go beyond the constriction region where actually the erythromycin then sits. But this is, of course, something we want to do, clearly. Right. I'm just wondering, shall we stop here and make a break, or shall we continue?
Yes, we can, I mean,
and a few questions, maybe.
You can decide if there are a few more slides that you want to take a pause at, that will be fine too.
All right. So since we started a little later, let's spend another 5 minutes or so, or 10, and then we make a pause, and then we are done with the ribosome story. So, okay.
That's
Okay, perfect. So now we switch gears. We still are with the ribosomes, but we switch gears a little. And we have a very close collaboration with Holger Stark at our institute and a number of other cryo-EM people, which actually do this nice cryo-EM work on ribosomes. As you might know now, the resolution of cryo-EM has, even for the ribosomes, surpassed that of X-ray structures, right? So that's quite amazing. And it's really amazing that people extract this information from these images, right? So these are the ribosomes here. And you get a resolution of up to 1.5, or even sometimes for other proteins below 1 angstrom with cryo-EM these days, thanks to new detectors and new refinement methods. Okay, so this is the images they start with. And you might know, of course, it's called cryo-EM for a reason. It's going to cryogenic temperatures before you do the pictures, because you want to preserve the structure and you want to reduce radiation damage by the electrons. And so what they do is they shock freeze the sample and then look at the structure, but not only at the structure, but also at the structure heterogeneity. So what you can do in cryo-EM, you can collect many particles and also not only get the average structure, but how the structure varies around this average. So every single particle is a little different, kind of caught in the act, a snapshot from the room temperature ensemble, where it actually moves and performs its function. And the idea is that you shock freeze it so fast that you just capture a snapshot for each particle of this room temperature Boltzmann ensemble, right, of the motion occurring at room temperature. That's the idea. And this is how they get what they call B factors. That's just a measure of how each single atom differs in the structure from snapshot to snapshot, if you want. And red means a lot and blue means quite few. Here's a cut through, so you can see on the periphery you have much more motion than the interior. That makes a lot of sense. And then here, looking at the surface, you see some parts which are particularly active. So some of you might know the L1 stalk, which is very active. That is one example which would be colored here in bright colors. So this is what you see in the cryo-EM. And of course, you can do the same thing in molecular dynamic simulation. That's shown at the bottom here. So you don't need to shock freeze, of course, in the simulations. You can just look at it at room temperature and record how much every single atom moves and do the same coloring here. For the cryo-EM experts, that's not an exact one-to-one comparison, right? So the B factors are a little bit different than what we do with the root mean square fluctuations in the simulations, but it's a good proxy. So we do have a quantitative, almost quantitative measurement. And it should agree, ideally. And it does, right? So we do have here, this part here agrees quite a bit. This part agrees, but not always, right? So you see here this part, for example, being highly mobile in the simulations, not so much in the cryo-EM, right? Also there's a very mobile part in the simulation, which is not there in the cryo-EM, not mobile there. Now you might say in the simulations, we have a limited time scale and some motions may be so slow that you just don't see it in the simulations, yet in the cryo-EM. Okay, that's a valid objection. But here it's just the other way around, right? So we do see simulation in the simulation motions which are not seen in the cryo-EM. And this shouldn't really be. And the question is, why is that? And that prompted us to ask an even more general question, namely, is it really true that the shock freezing is fast enough to really carry a snapshot? Or can it be that during shock freezing, something happens, restricting the motion? Yeah, let me just remind you what happens during shock freezing. So the sample is on a grid, right? And you have these tiny little holes, and the solution with the contained biomolecules span these little holes. That's a few from the side here. And it is this thing which is plunged into liquid ethane as quickly as possible. And if I ask my friends, they will tell me it's about a millisecond it takes for the whole thing to submerge, just with a spring-loaded gun pushed into the liquid ethane. But if you look at it from the perspective of a single ribosome like this dot here, it may be a bit faster. Reason being, as soon as the first hole here gets submerged, it starts freezing. And it doesn't have to wait for the whole thing to submerge. From the perspective of a single biomolecule, it just counts how long does it take after this water film here gets in contact with the liquid ethane until the heat diffuses to outside and the biomolecule gets cool, right? So how long does this process actually take? And this is the question we asked first, which then helped us to study this question here. And to make a long story short, we found it takes just a few microseconds because the water film is so thin. We don't know precisely. It could be 100 nanoseconds. It could be a microsecond or a few microseconds. That very much depends on how thick the water film actually is, which nobody knows. It's very hard to measure. But the effect which we expect is the following. So this is now a very schematic drawing of the very complex energy landscape in which proteins actually move. That's the landscape which governs for the experts, the free energy landscape, which governs the motions of the protein. And at room temperature, they jump from one to the other minimum, many minima. Again, some of you might know the Frauenfelder picture. That's what I'm showing here. Really many minima separated by a hierarchy of energy barriers. Smaller ones, higher ones, the higher ones being traversed slower, the lower ones being traversed faster, but the whole ensemble moves back and forth between these many minima. That's how protein motion comes about. That's the statistical mechanics picture of it. Now, if you cool down instantly, of course, the whole thing is shock frozen. Nothing moves anymore, and you really get a valid and accurate picture of this ensemble in the cryo-air. That's what people dream of. That's what they want to achieve. But again, it's microseconds. It's not zero. It's microseconds it takes. And it may be that during that time, the lower barriers are still overcome. And the whole ensemble shrinks towards the lowest minimum from the higher minima here, overcoming these barriers, the lower ones, maybe not the higher ones. For that, the time may be too short, but the lower ones. And as a result, you will see that the width of this ensemble in red here at room temperature gets a little smaller, gets a little constricted, narrower at 90 Kelvin as a result of this not infinitely fast cooling process, right? And so in more detail, the question which we had was, how fast is the cooling? Actually, I told you that already. And the essential question, how much is preserved of the structural heterogeneity which we find at room temperature, of the physiological motion of this machinery. And as a corollary question regarding the kinetics, how high are the energy barriers actually which are still overcome during the shock freezing as opposed to those which are too high to be overcome? Okay. That is just a short graph supporting what I was saying here. That's the setup which we do in a very simple continuum calculation that has nothing to do with molecular dynamic simulation. It's just solving the heat equation for a slab of water immersed into the green ethane. And what we find, as I said already, depending on the size of the water slab, the thickness of the water sheet here with the embedded ribosomes, we get such a cooling curve. And we reach 150 Kelvin at about microsecond timescales, maybe 100 nanoseconds. And at 150 Kelvin, not much happens anymore, as you can see also here. So to answer these questions, now, of course, what we do is molecular dynamic simulations. So rather than just carrying out the simulations at room temperature, we thought, why not just simulating this shock freezing? Microseconds, yeah, that falls into our comfort zone, into the time scale which we can simulate in a couple of weeks. So what you can see here is lowering the temperature to 90 Kelvin from 300 room temperature during the simulation. This is just a zoom here into the structure. And if you look at these side groups, very nicely you can see how they kind of freeze out. They stop moving and freeze out in a certain conformation. And if you do that again and again, and this is exactly what we did, then you will find them in each attempt in a slightly different end configuration frozen in. We did that to get better statistics. Now, in 41 cases, so we took a long room temperature trajectory, and at random points in time, more or less, we took a structure and shock freeze it, right? These are these indices here. And we did so in each case very slowly as we could afford, and insanely fast, just to get a comparison how much this cooling depends on how fast you cool. Yeah? And this is now probably one of the most complex graphs I'm gonna show you. No worries. What you can see here is in the columns, each column here is a different cooling time. Yeah, those are the very slow cooling simulations, and those are the insanely fast cooling simulations. Now, in each column, you have a time scale, and this is normalized now to the total time during cooling. So these are 128 nanoseconds here and 0.1 nanoseconds here on the left side, and everything in between. And the curves show now exactly what I have shown, symbolized before with this graph here. They show how much the width of this ensemble narrows down, right? So we have our 41 end structures, and for the experts, we calculate the root mean square fluctuations of the whole thing for each atom and average, right? And that gives us eventually the value shown at each time in the curve. We do that during the cooling process. And the grays our variation which we observe due to the fact that we have only 42 ensemble. That's done with bootstrapping for the experts. And I guess you will agree with me that each single curve here actually decreases quite nicely. Oh yeah, I forgot to mention what the rows here are. So those are quantiles, right? So those are the atoms which show the 20% atoms which show the largest fluctuations, second largest, and the least fluctuations. So those would be the small fluctuations, which are typically fast, and those would be the large one, which are typically slow. And you can see already nicely that the effect of the slow ones is not as pronounced as the effect of the fast ones. The fast ones, remember, are the ones which overcome low barriers, and the low barriers are the ones which we suppose should be overcome still during the shock freezing, whereas the high barriers probably not. So we see the largest effect here. Can we rationalize that in terms of a simple model? And here we have now one of those top-down models you are being used to. We just say, okay, we have a certain structural heterogeneity before and after cooling, and we have a barrier in between, and we use the height of this barrier and the rates and the distance between the two ensembles and the scatter of these ensembles. We use those as fit parameters, which we extract from these curves. And the result of this simple model is here now superimposed as the blue curves. And I hope you can agree that the blue curves describe the whole thing with just 5 fitting parameters pretty well. And so we would maintain that a simple two-state model, like, again, if you know a little bit about neutron scattering, that's what the models are to interpret neutron scattering data, actually describes our cooling process pretty well. So we think we understand what's going on in terms of kinetics, in terms of free energy barriers during this cooling process. And one of the things which we extracted from that data, 2 important numbers. One, about 20 to 30%, that's what the result of this fitting model, of all the fluctuations are lost. That's what we see in the simulations, which is pretty good news for, if you wish, both sides, right? Our friends from cryo-EM, they rely on not too much being lost. And you could argue that 20, 30% is not so much being lost. So the large fraction of the room temperature motion, they capture, at least for the ribosome, they capture quite reliably. That's the good news for our friends. For us, the good news is, first of all, we know that now. And second, it's not 100%. So for us, it means we can help. We can now use the simulations and rescale back what the room temperature ensemble, given the cryo-EM ensemble, what the room temperature ensemble should have looked like after this shrinking process to result in the cryo-EM ensemble. So we get a better estimate, a recalibrated estimate, if you wish, of what's going on at room temperature, at physiological temperatures, given a reliable cryo-EM ensemble. And again, I was mentioning this Frauenfelder picture with different barrier heights. And on one of the side results, that was the third question we had, was, okay, about 10 kilojoule barriers are still being overcome. And then, of course, now we are starting, that's not yet finished, but we are starting to look into what motions precisely are those, what degrees of freedom precisely are those, which are not frozen out, but which are still equilibrating during the shock freezing, right? And those are generally the ones with the high 10 kilojoule barrier, but what are those? And I mean, it's not so hard to guess. That will be side group motions, etc., as opposed to, for example, if you know the ribosome, the 50 relative to the 30s subunit rotation undergoing during elongation. This motion will certainly be frozen out and reliably be captured by cryo, as we know. So this is what we learned from these type of simulations. I would believe that these were the first shock freezing simulations carried out ever. And we did that for the ribosome because we have the best cryo-EM data. Of course, now we are gonna move on and do it also for like proteins, protein complexes. The proteasome would be one of our favorites and so on. So I guess that's a good point to stop for a break. And I'm happy to answer questions if you want. Yes. Okay, we have a chairman here. I shouldn't take the role of the chairman here.
Hello, thank you. It was a nice talk. The second, I have two questions. I'll ask about the second part first, since it's fresh. So if you think that the populations are getting redistributed, can you actually calculate the actual population at the of a particular conformation? I'm talking about because I want to, many times we get low, if you do classes, you get some classes which are low populated and they could have been readjusted during cooling. So if we could populate, we sometimes do 3D variability. Instead, we could actually know more about that population if we could know the percentage drop in the population.
Right. Yeah, thanks for the two questions. I'll start with the first one. Can we calculate the population? As physicists, I would call it an ensemble, and an ensemble is a high-dimensional beast. So having only 41 samples, simulations, we get an ensemble. We get 31 different structures, and you can calculate whatever you want from the distribution of those structures. So the answer would be yes. Do we capture reliably the whole and cryo ensemble with 41 structures? I'm not sure. It's good enough to calculate what I've shown you, but if you want to really calculate conformational densities, that would be a long stretch probably. So that we can't. Second question, can we calculate the population as they change during cryo-EM? Yes, that is what we can do. And this graph, which I was showing, captured the shrinking on this population. What we have not yet done, but we can do that, of course, look at specific conformations captured by cryo-EM and see how they change during the simulation. This is what we can do. I would suppose that what you see, but this is a wild guess or speculation from what I saw, that the populations will change by a factor of 2, something which you might see at 20% of the time may change to 10 or 40, depending on where it is. Not much more. That would be my ballpark guess of what you would observe. But yes, that is what you can do. And one restriction I have to say, one caveat I have to mention, that would assume that we see the full room temperature ensemble in the simulation. And unfortunately, we don't, again, because our timescales are too short.
Yeah, the first part, about the ribosome stalling, I have Again, two questions.
Yes.
One is that, you showed that there is a change in the tunnel when
Yes.
erythromycin is present.
Yes.
Now, can we relate it to exactly how stalling has happened, because that's stopping translation? So allosteric change because of the tunnel width is actually stopping the translation.
Let me try to go back a few slides. That's essentially what I tried to communicate, right? So what we see is that this part of the tunnel goes to the left, moves to the left as soon as you add the erythromycin, and really drags a charged amino acid with it. And the distance to the growing chain increases. That's
no, but it is actually stopping translation. So it has to go back to the PTC and do something such that no more translation happens. Is there a readjustment at the catalytic center? That's what I'm asking. Or there's the allosteric change does not reach the catalytic where the tRNAs are actually the nucleophile which is in the PTC.
Yes. So the stereochemistry for the nucleophilic attack is perturbed, and the distance between the incoming amino acid, which is about to attack.
Sorry, I missed that.
Does that answer the question?
Got it.
Okay.
Got it.
Great.
And the other question is, what kind of structures will not cause stalling? So if you have all antibiotics must not stall, right?
So, can you
all the time, if you put any antibiotic in that position, it will not stall. So what is it, the position that matters or the structure that matters?
In this particular case, it's the charge of the amino acid.
It's only the charge of the amino acid.
So that if you want the type of amino acid, that makes it sequence dependent.
So you can put any antibiotic, erythromycin or anything else.
No, that's only true for erythromycin. If you have another antibiotic, the mechanism is a different one. For example, we looked at one antibiotic, keromycin, which has a totally different, it affects the incoming tRNA and actually avoids the tRNA to adopt the position which it needs to place the amino acid just in the right position. So it doesn't change the wall, but it has another allosteric interaction which prevents the tRNA from adopting just the right position. And it's really fraction of an angstroms which make the difference. So it is still in the binding pocket. Cryo-EM people would be happy and probably wouldn't even see a difference, but the minute distances here also cause stalling. That would be one. A third stalling mechanism which we looked at was that in this particular case, it's self-stalling. It's stalling without an antibiotics. Just a particular sequence adopts an alpha helix here right before the constriction region. So that is quite unusual that you see secondary structure in the tunnel, but this particular sequence is so alpha helical prone that it forms transiently an alpha helix already here. Now, again, you might think, okay, that stops everything, that clogs up the tunnel, jams up the tunnel. No, it is this alpha helix which acts back again on the active site, on the PTC, and in this case, rotates this amino acid such that a single hydrogen, gets into the way of the nucleophilic attack. So in each single case, we see a different mechanism, but the common denominator is it is an allosteric interaction quite over a distance, and it perturbs the active site, the stereochemistry of the active site. That's what we see all over again. Thanks for giving me the opportunity to lay that out a little bit and sneak a few minutes.
Hello, I have also two questions, but I will, instead of time, ask only one. So with respect to the simulations of, pushing the peptide through the tunnel. So you mentioned there are lots of movements of the ribosome during elongation. Rajyaguru is one of them, but there are a lot more, along different axes, the rotations. So do you consider these movements in your simulations? Because I think they may play a critical role for this pushing, maybe something like squeezing up, the tunnel to push it up. So do you consider these in your simulations? Thank you.
The short answer is yes. And actually, you saw the name. Michael Koller was one of the ones, your colleague who did one of the simulations at the time when he was. We have two options, and we do both. One is we take the whole ribosome into the simulations. In this case, we consider all motions of the ribosome. And then for production, and this we do as a test, and for production runs to get better statistics, we cut out the whole tunnel quite generously. It's quite a large fraction, but still the simulation system is 10 times smaller than the whole ribosome, which gives us 10 times more performance. And we construct the tunnel, the walls of our box such as to reflect the motion of the ribosome at the end of our simulation box. And we compare the fluctuations which we see with the simulations of the whole system to make sure that we capture all the motions. And that's why I'm quite confident that we actually include all the motions. And I fully agree with you. They are super important. If the whole thing wouldn't move, I don't know yet, but I would speculate it wouldn't translate through the tunnel. I'm pretty sure. Mm-hmm.
Can I?
Look. Yeah.
Ensemble from the cryo-EM data, can we fasten up the MD simulation process by using those ensembles from the data? I mean, if we don't have the models of those ensembles, but there might be maybe using 3D variability, which Ruchi was talking about, can we extract those ensembles and fasten up the MD process?
I'm not exactly sure if I understand the question. Can we extract the ensembles from the simulations?
From the cryo-EM data.
From the cryo-EM data. Yes, they can extract ensembles from the cryo-EM data. They don't need to be fast with the data acquisition, right? They just need to be fast with shock cooling. Once it's shock cooled, it's frozen and doesn't move anymore. And then what they do is they take these images, which I've shown you, right, where the ribosome is hardly visible, and they sort all these particles into individual orientations first, but also into different conformations, structures. And after they sorted it into different conformations, they average each sub-ensemble into a certain conformation. And that's how they get different structures out. Does that answer your question?
No, but I think it's implying that from cryo-EM, if you can get certain snapshots,
yes,
but not the complete thing, can you then speed up your simulation with those as
speed up?
Some sort of, yeah.
Is that the question?
What you're asking?
Can we speed up?
You're taking 2 weeks to do these simulations.
Yeah.
Will having certain intermediates along the way.
Yeah. Okay.
Will be able to speed it up.
Okay, thank you. That clarifies it. Yes, the clear answer is yes and no. So yes, it helps us, but no, not directly to speed up. And let me explain a bit. So what you probably want to know is you have one structure from cryo-EM, you have a second structure from cryo-EM. You want to know how the system moves from here to this structure. How does the structure change in detail? Maybe what is the energy barriers obstructing the structural change or slowing down the structural change? What happens in detail? There is, and if the barrier is low enough, you see that in the simulation directly, right? But if the barrier is higher, you don't see that. And this is probably what you mean by speeding up. Now, there are methods to push the system over the barrier. If you want to look it up, umbrella sampling would be one of those methods. That's one of the acceleration methods to push it over the barrier. If the structures are not too different from each other, that works pretty well. And then you don't need any help. You can do it from the simulation and see how the system behaves and traverses the barrier. However, if the barrier is too high or the two structures are too different, that becomes less and less accurate and takes more and more computer time. And there, that's my yes part to the answer. If you have intermediates from cryo-EM between the two big different structures, then that helps this method. It doesn't help an unperturbed free simulation because I wouldn't know how to do it. But these artificially accelerated simulation, for those it helps a lot. And this is exactly what we have also done. We have pushed the system from one intermediate to the next and thereby followed the whole reaction path. There is one caveat though, which I also need to add, which is the cryo-EM ensemble doesn't have time information, right? If we have a number of structures from cryo-EM, we don't know if this is the right sequence in time. It's just an ensemble, right? It's not a time sequence. So you need to add an assumption, which is similar structures are subsequent in time, right? It's unlikely that the system will jump between very different structures in a short time scale. It's much more likely that in reality, the system will move from one structure to the most similar one and then to the most similar one and so on. But this is an assumption we have to make, which we cannot read out of the cryo-M. I hope I could answer the question now. Okay. Thanks for the question.
I have not question, but I would like to have your comment. So structural heterogeneity is significantly reduced at cryo temperature, right? Than room temperature. So if we are using the cryosol structures for like structure-based drug design or biological outcome, how relevant it will be?
How reliable will the simulations be?
Not simulation, but the structure solved by cryo-M used for structure-based analysis or structure-based designs.
Yeah.
So,
okay. Okay. I can't tell really, because I don't know the room temperature ensemble. So, of course, from what we know, these structures have been used for drug development and quite successfully so. So my take would be that the motions relevant or the structural heterogeneity relevant for drug design, for example, to see where the chiromycin or where the erythromycin is actually located. I think that is quite reliable because there is not much structural heterogeneity associated with it. What will be less reliable are the motions, the more large-scale motions, which are overcome so slowly over the barriers. That would be my wild guess. And from the simulation side, another question would be, are the force fields, which I was mentioning the force fields in my introduction, right? That's one of our weak spots. Is the force field good enough at cryo temperatures? I would hope so. And we have done quantum mechanical calculations to support that. But I clearly have to say the force fields are optimized for room temperature and not for cryogenic temperatures. So it may be that the force fields are a little off. Yeah, that would be another point of argument.
Thank you. So me again, I'm sorry, I have to ask the other question because it's basically a follow-up to that question. So the fact that, you can lose, so usually you lose, especially with ribosomes, roughly 20% of different populations of the ribosomes.
Yes.
So I think if you collect large amount of, like enough amount of particles that you can really produce lots of different populations.
Yes.
So then you should mitigate this problem, right? So then you should have enough images that could create the movie. The reason I'm asking because, of course, Christian Spahn from Charité. So he was 2 weeks ago, he was presenting, his student was presenting a collection of, I think, 30 or 35 different states of the elongating ribosome. And I was thinking like, oh, come on, this is crazy. Why? Now seeing this, he could have, by this large sort of set of populations, he could have circumvented this problem, couldn't he, in a way, right? Because then he should have even these populations that represented this high barrier that has to be crossed.
Yeah, I agree. I wouldn't call it circumvent, I would call it mitigate maybe. So the more images you get, the more classes you can split your set up into, the more likely you will discover those. Nevertheless, you will discover those with the wrong population, right? The population will still be off. You will find the structures, or you will push the envelope a bit, right? So there will always be structures which will not be able to find no matter how many images you take. Unfortunately, sometimes those are pretty important, and that is where also we come into play. We are collaborating with Christian on essentially that set of data on that one. But yes, that the more images you have, the better you are off, and that mitigates the problem.
And proceed to the second half.
Shall we proceed?
Yes.
Okay. No bio break or something? No, you decide.
Along with the question and answer session. Do you want to take a break? And then
Not me, but
We have another.
in fifth, we actually delayed.
yeah, we have 1/4 of an hour left, so yeah. Okay, okay. Yeah, so as this was the first of my stories, I'm a bit pessimistic whether we will really manage to get all 4 of those. So let's see. Let's see where we are. What I would suggest is to move to point 4, because that is, I guess you guys are interested also in structure determination methods, maybe not so much into details of statistical mechanics of solvation. Is that, I see a bit nodding here. So my suggestion would be that I speed up, zooming to my last story. If you give me a second to move forward, where I would like to expand a little bit on an alternative to cryo-EM. And that is actually the story of Steffen Schulze, mostly in my group. And since a couple of years, we are heading towards single molecule X-ray scattering. And let me explain a bit what that means. So it's no cryo-EM anymore. We are going back to X-ray scattering. And does it work? No. Yeah. And the idea is the following. We have, since about 10 years, super high intensive free electron lasers available. One is in Hamburg, another one is in Stanford. We have in the UK a diamond laser facility, I think in Switzerland. So we have a couple of these super large, super expensive accelerators, which accelerate electrons. And if you shine these electrons through a Wiggler, they emit super focused, super intensive X-ray scattering, X-rays. And they are coherent. And that means you can do crystallography with them. And this is done for 10 years. What people do is they throw nanocrystals or microcrystals of proteins and biomolecules into the beam and get nice structures from that. The advantage being that it's easier to grow a small crystal than the large ones needed for conventional crystallography, right? So this is already state of the art and done. I think it would be nice, and this goes back to such groups, to a suggestion by actually Richard Neutsche and Janusz Hajdu and Henry Chapman also a little later. So they were the first to suggest, why can't we drive it to the extreme and scatter on single molecules, single proteins, single ribosomes, foregoing the need to crystallization, right? So you don't need to crystallize anymore, which would be nice, right? Also, you would need much fewer samples, much sample size, and you don't need to crystallize. Also, if you have a crystal, you always average over many specimens, many proteins. And if you look at a time-dependent process, you average over many motional states, yeah, these different conformers, which we were talking about before. If you do single molecule scattering, you get a time snapshot, and you get access again to the whole ensemble and maybe even to time information if you're able to trigger a process. Imagine you trigger a process here with a laser, and then a few picoseconds later, you flash the X-ray beam on it and then record a scattering image. So you can get time-resolved movies of protein motion, like we see in the simulations, but now measured. That would be the ultimate goal. So this is why I'm quite excited on this type of experiments. And the width of such a pulse, of a laser pulse, is really super short. So we are right in the time scale of our integration time steps, of the fastest motions of the proteins. So you really get a super sharp, super short exposure picture. The drawback of the whole thing is the intensity needs to be super high. Why is that? Well, if you just shine light on a single molecule, you wouldn't expect many photons to be scattered, right? It came like 10 years ago, I learned, and it came quite of a surprise. The real trick of X-ray crystallography is not that you have these nice Bragg reflections and so on. The real trick is that you have a super amplifier by an Avogadro number of molecules. So that is the trick, where one molecule just scatters a little, 10 to the 20 molecules scatter a lot. That's why you do crystallography. And so here you can expect a super small scattering cross-section, and with conventional light sources, you wouldn't see anything. But with these high intensity, you do. And the drawback, however, is that the intensity is so high that it shoots out half of the electrons of your whole molecule immediately, right? And as a result, all the chemical bonds are destroyed and the whole thing explodes in what's called a Coulomb explosion. But the pulse is so short to outrun the explosion. It happens only after the exposure, and therefore you get a nice image, or at least some scattering information of your molecule. That's the idea. It's called serial crystallography, right? It is a little bit, if I would use a nuclear bomb as a flashlight to take a photograph of you, if I have a short enough exposure, it will work, right? You would probably not be able to see the image later on and admire your face, but it would take an image, right? It's about the same thing here. I guess that is why I need a security declaration and a special visum to come here, because I say these things. All right. Nevertheless, what you get is just a few photons. You don't get nice Bragg peaks. First of all, because you don't have a crystal, and you may know that the peaks arise just from the periodicity of the crystal. There is no periodicity in a single molecule, so you don't get Bragg peaks. What you rather get is a continuous image, a continuous scattering image, which theoretically would look like that, as opposed to what you get from a crystal, all these Bragg peaks. So there's no peak. But this image would be perfectly suited to calculate back by inverse Fourier transform. Forget about the phase problem for a minute. It would contain enough information to get the molecular structure out of it. So far, so good. But again, I said we only have a few photons. And so each single image looks a bit like that. And that is a bit of a problem, right? So you can't extract any information from the single image on the structure. Well, at least it seems so. And if we go back to cryo-EM, I would say there's a lot of noise in cryo-EM when it comes to structure, but there's even much more noise in these images. So that's the mother of all noises, if you want, right? So the challenge now is to yet get the structure out of these type of images. And the advantage which we have is, of course, that we have many of those images. So these free electron lasers have a repetition rate of 25,000. So you can take actually 25,000 images a second, which accumulates to many millions of images, which you can accumulate in a few hours, over a day or over a night. So we have many of them. One of the problems, of course, also being that the orientation of the sample is unknown. If you do a crystallography, from the pattern, you can deduce the orientation, and you can superimpose all the individual scattering images, which you can't do if you don't know the orientation. So that's an additional complication. And plus, I have entered red dots here. Those are the coherently scattered photons in this sketch. And the black dots are background scattering, which you can't distinguish on the detector from the red ones here. So you also have to face considerable background noise, which makes it, as it seems, very hard, right? And actually it is. And there have been a whole lot of different approaches to trying to get actually a structure out of it. And one of the questions we also had was how few photons we'll do. And this is just, again, for the experts. There have been, over the past 10, 20 years, a whole lot of suggestions how to extract images from that. All share in common that they need at least a few thousand photons. If you really drive it to the extreme, a few photons, a few hundred photons, to be able to extract it. And the reason is each single method of all of those try to orient the sample first, try to determine the orientation of the molecule first. And then, of course, you can superimpose all images and you get a Fourier density, and from which you can go back and calculate the structure. So the question is, can we circumvent, can we avoid the need to orient each single picture? Can we get along without knowing the orientation of the molecule in each single shot? And it may sound quite impossible, but a few years ago, we have shown that if you actually calculate correlations, and I'm not going into detail which correlations those are, but they are correlations which are independent of the orientation of the molecule. And if you calculate those from your set of images, then you can get back the structure. We have shown that in this publication here. And the surprising result is that 3 photons per image actually suffice, right? If you just have 3 photons per image, then you can get the structure, which sounds quite incredible, but actually we have shown that works. What I would like to show you now is how we can get better, because these correlations, they throw away a lot of valuable information. And if you have very sparse data, you really want to make use of all the information available. And what we now recently did is another approach, which actually makes use of all available information. And that's a Bayesian approach. And I would like to use this opportunity to really advertise Bayesian approaches. If you haven't heard about them, yes. Who has heard about them, yes, so far? What is about Bayesian approaches? Okay, so probably it's a good idea to advertise that a little bit. So what a Bayesian approach, it goes back to a guy called, well, Bayes, living in the 18th century, coming up with this formula. And I adopted this formula now just to the current situation of our structure determination problem. And that's the following. What you want to calculate is, given all those images, that's conditional probabilities, right? Those you know, right? So given all those images, all the data, the whole amount of data at once, as one chunk, right? Conditioned under this data, what's the probability of all possible structures you can think of, and then you pick the most likely one. Sounds simple, right? Except that this you cannot calculate directly. Impossible. It's a so-called inverse problem. Nature always conspires against us that the interesting problems are always the inverse ones, where you need to back calculate. We only can calculate forward. What do I mean by that? We can, of course, simulate the experiment. We can say, let's assume for the moment that we know the structure. That's this probability here. Given we know the structure, then we can calculate forward what the experiment would look like. We simulate the experiment. That we can do pretty well. Maybe expensive, but we can do that pretty well. I should mention what I'm talking about now has nothing to do with molecular dynamic simulations so far. It may enter later, but not today. So this we can calculate. But I very, very much hope, I saw that there's probability theory also present in this center in your introduction. I very much hope that you know that this is different from this. If you ask people on the street, they don't know that, right? You can't switch the condition and the argument of the probability. It's a different number. It gives a different result. I hope you know that. If you don't know that, if you're surprised by that, please, please read it up. So important. Also to biologists, really. So you can calculate that, but not that. But Bayes tells us, quite simple, that if you multiply that with what's called the prior, which is the probability that this structure occurs before you take up the experiment. So this is all you know before you make the experiment. Your background knowledge, your prior knowledge about structures. Think about, for example, the PDB database. You know what all the structures in the PDB database look like. Think about AlphaFold. You know what AlphaFold suggests you. It may not be the correct structure, but it's a prior knowledge you have about the structure. That is the probability which goes into there. And if you take the product of that, if you look it up in Wikipedia, you have to divide it by a denominator, which is quite clumsy. I omitted here to make it a bit more simple. That's why I have to put the, replace the equal sign with a proportionality sign. But that's the essence of Bayes. If you know a prior, if you can calculate that, then you can calculate what you really want to know. Given the experiment, what is the structure? And this idea we implemented, and you don't need to read this math. I'm showing you that just that you see that we know what we are doing and what we have to do. This is just the implementation of this likelihood in mathematical terms from the given structures. That is something that can be calculated. It's not so hard. And that gives us the structure. I'm skipping that, and I'm showing you just a way to sample the posterior. I should probably say one more word on that one. This thing, as I said, is hard to calculate, but it can be sampled. What you do is you modify your structure with a Monte Carlo approach, tiny little changes on the structure, and thereby you sample all possible structures. And at the end of the day, you get a whole ensemble of structures with weights, probabilities attached to each of them. And then you can calculate, for example, a maximum or an average structure and so on. So this sampling, that's the hard part. And that is what we solved for medium-sized proteins. And I'm just giving you a visual impression of how this sampling actually works. This is the electron density of our small protein, which you took as an example. And these are all the atomic positions of our structure, which still are, as you can see, wiggling around quite a bit, highly undetermined. But then we crank up the resolution in the simulation and compare more and more detail to the experiment. And you see more and more detail emerging from the electron density of which is actually what we fit to the data. And at the end of the day, you see a high resolution structure with all the atomic positions quite accurately positioned. This is now, I should say, a synthetic example, right? We generated the measurements. People haven't been able to carry out these measurements yet, but we better prepared. And this is what it looks like. And this is just to show you, yes, we can actually solve a structure given a million or so of those images, which I was showing you before. And we arrive at a pretty high resolution. I think in this case, it was 2 angstrom. That's misleading. That's just a smearing out, the reduction of the resolution. What we actually get, here you can see again the reconstruction and the reference, virtually identical. And those who are into cryo-EM, they know that the Fourier shell correlation coefficient, that is the golden measurement for resolution. And measured with that, you get it about, depending on the protein, 2 to 1.5 angstrom resolution. So this is what should be possible with this kind of experiments. And this is a kind of proof of concept. And in the last 2 minutes, is that okay? I'm just showing you that it actually works with real data, which requires a lot of experimental uncertainties being implemented in the likelihood function, which is another advantage of the Bayesian approach. We can systematically implement all experimental uncertainties and noise and background noise, weird detector shapes, different detector efficiency between the different pixels of the detector. We can implement all that into the likelihood function and then get a rigorous implementation of all the experimental uncertainties and errors, as opposed to the previous methods where you just have to hope that your method tolerates the errors good enough. This is just to give you another impression how these images typically look like, right? So only the red dots are signal, the other ones are just noise, and you have to make sense of all those images. So we did that now, and I'm skipping forward. Let me to that one. To real data, this is one of the real data we were using. This is not yet a protein. As I said, people have not been able to do experiments on proteins yet, but they have been carrying out experiments on viruses, way larger. And these are the typical images they get. Way more photons. So here the conventional methods, they work. That's why they restricted themselves to viruses. We said, let's make our life harder and test how far we can push that and downsample these photons to just a handful and then use our method to extract this structure here. And this is what we get from our method. It's very closely, in part even better what they extracted with the many photons available using established methods. And we were using 1,000 times fewer photons per image. So that also shows that even in the very low photon range, which we would expect for small molecules, our method works here. And this result has now triggered 3 experimental groups in Hamburg and in Stanford to actually carry out the experiments we are dreaming of. And we are really hoping to get results. And I'm showing you just one unpublished result of a sample which we have provided. And this is DNA origami. So this is now a real, if you want, molecule rather than a virus. It's way smaller than a virus. Here you can see the scale bar. And this is the particular origami we choose. There we know the cryo-electron structure, which looks like that. And this is what we get so far from our reconstruction. That seems to be a bit disappointing. But mind you, a good friend of mine, Holger Stark, in fact, who is into cryo-EM, he said, well, Helmut, this is just where we were in cryo-EM 20 years ago. And I said, yes, exactly. That's where we were. And look where you are now in cryo-EM. I think there's a bright future for this method. And mind you, we don't need to shock freeze. We get the real ensemble and we get time information. And I think although these accelerators are even more expensive than cryo-EM, I would think this is a nice alternative structure determination methods. And I'm quite happy that I'm part of this endeavor. And I'm probably skipping forward to my last slide because my time is over. And just summarize what we have probably learned now from this single molecule X-ray scattering, forget about that, experiments. I think we have learned that Bayesian methods, and we have seen that over again. If you look through our publications, we use Bayes all over again. To the biologists, I would propose really forget about hypothesis testing. It's not the real thing. If you look into the literature, hypothesis testing, I'm a bit challenging now, but just to make you a bit think about what you're doing when you do hypothesis testing, you're answering the wrong question. Think about it. You're just answering for drug testing, what's the p-value if the drug would not work that I get the result of my trial that I actually get. If my drug would not work, what's the probability that my experimental result comes out? Remember what I've shown before? It's the wrong question. It's a good answer, but to the wrong question. What you really want to know is, given the trial output, what's the probability that my drug actually works, right? That's what you want to know. And your hypothesis testing does not answer that question. Think about it and think about p-values. And I think Bayes is much better. And it's a historical accident going back to a guy called Smith in the 20s who actually promoted hypothesis testing for a wrong reason. All right, so I think we were able to show that at least in principle, we should be able to achieve below 2 angstrom resolution with this type of single molecule X-ray scattering experiment. I mentioned that there's a very systematic way to include all experimental drawbacks and sources of systematic error and noise into this, which is not possible with other methods. As a result, we needed 1,000 times fewer information because Bayes is targeted towards extracting maximum information. So far, the best we achieved in DNA origami in real data, real experiment is 9 nanometer resolution. Again, that's where cryo-EM was like 20 years ago. Remember these blobby images of ribosomes, which we got in the first place. I didn't expand on it, but you also can refine ensemble, very much like you can do in cryo-EM. You can also do with ensemble refinement. And I didn't write it on the board, but what we are doing also now is to try to extend and modify these methods to make them usable for cryo-EM, because there's mathematically a lot of similarities between this, what I've shown you, and cryo-EM. And so that also should work for cryo-EM. And with that, let me just conclude that when it comes to simulations, I think we have come a surprising history and way, starting with solving a Schrödinger equation for very simple systems like a water molecule, or the Nobel Prize winning simulations of Martin Karplus and others of the first proteins in the 70s. Proteins in their full environment, like beginning of 2000s, and machines coming up, viruses or so, larger and larger systems. Today, with coarse-grained simulations, we are aiming whole cells. So we are approaching this size. I haven't shown you microtubules, whole microtubules, which we have done recently. Those would be vesicles, which are on our to-do list. And eventually, I don't know where that will lead us. But I think from our perspective, we live in very exciting times. I'm really privileged to be part of this wonderful history, to drive and to push the envelope of simulations to longer and longer timescales, to larger and larger simulations. AI will be an integral part of that. And with that, I'd like to thank you and those guys who have done all the work in my wonderful group here. Thank you for your attention.
So just a quick question. Would you consider virus already as a form of?
I don't care.
I'm asking you.
Yeah.
It is technically a crystal, right?
A virus is not, a crystal, not alive, you say. Okay. I wouldn't say it's a crystal. You can crystallize a virus nicely, but it does have a symmetry, yes. It would be a kind of a rotary crystal if you want. I'm a bit skeptical about questions. Would you call it a crystal? you give me the definition of a crystal, and I tell you whether I call it a crystal. Other than that, it's semantics. By the way, you biology friends, the same, are viruses alive or not? I thought you would ask that question, and I really don't care. It's semantics. It depends on how you define life, and then it becomes a boring question. And I just would say viruses are super exciting, totally irrespective of whether you call them alive or not.
I accept the challenge because
Okay.
my research is hypothesis driven, obviously.
Yes,
Our
good.
research. So, you know, I understand that you can do all these, you know, simulations or calculations for isolated systems, you know, for single proteins or even molecules like the ribosome or part of the ribosome.
Yeah.
But if it comes to a molecular mechanism that involves signaling pathways, RNAs, proteins interacting with each other, you know, cascades and stuff like that.
Yeah.
I cannot imagine how you couldn't live without a hypothesis that you are trying to prove by doing experiments, getting false results, positive results that push you forward. In this way, maybe I'm not the mathematician, you know.
Yeah.
But I'm just kind of lost in what you say because I don't think that it will work for my kind of research.
Yeah.
That's the challenge that I accept.
Yeah. Okay, great. Challenge accepted. So first of all, although in principle you can conceive of simulating a whole cell with molecular dynamic simulations, with current technology,
It's a biological system. So many things that you cannot.
yeah.
Some experiments work differently.
Right. So now that's a different challenge. It's not the hypothesis testing challenge anymore. It's two challenges you gave me now. So addressing that one. If you were able, for example, now I'm really speculating and hypothesizing. If you were able, for example, with the FXL experiments, to measure the atomic positions of a whole cell, all of them, then I would claim, and if you were able to have a powerful computer to do the simulations, which we are far from having, just to be sure, but then I would claim this simulation would capture the correct development of a cell, right? But that's a belief. Yeah, you have to put it into water and you have to feed it and all that. But sure. But that's so speculative. That's just on the level of believing, and we shouldn't remain on the level of believing in science, right? So okay, so that is a thing. Of course, in physics, always we are working coarse graining our way up. We are also not describing the gas in this room atom by atom. We describe it by the laws of gas, right? So and since 100 years. And the same will be true for cells. So we will be working our way up. How will we do that? I don't know yet, but there are indications. So for example, people are actually working towards a whole cell model, at least these super tiny cells by Craig Venter and so, which are way simpler, with coarse-grained method, not atom by atom, but at least with a coarse-grained simulation method. And I'm not part of that, but they are working towards that. Whether it will work or not, future will tell. Now, you were talking about signaling pathways and stuff like that. That's a different story because one of my stories I could share, but haven't chosen to, is about Markov models. And signaling pathways you can describe with Markov models. So the idea would be to get the individual interactions, like binding affinity of a certain, I don't know, P53 molecule to different substrates. You can get that either from measurements or from this type of simulations, or ideally both. And that you can feed, and there comes Bayes into play again, into Markov models. And the Markov models can show you how these signaling pathways work. We are also not yet there, right? So we are working towards it, but we are not yet there. But you were asking about the future, and I would guess that's one way to work our way upwards to signaling pathways. That's not a whole cell, surely not, but it's a different aspect, which hopefully will become computationally tractable in the future. That addresses your second challenge. I'm not sure if I could talk eternally on that. But, and I could address the hypothesis testing, but in the interest of time, let's push that to the coffee break, maybe.
Yes.
Yeah.
So in the interest of time, we'll stop the discussion at this here, but you all please continue the discussion over coffee and tea.
I'm around.
Yeah, he's around. And we'll also now first assemble for the group photograph, followed by the she'll guide us. So I think it's